Hi everyone, welcome back. Second topic of the course. I hope you took a little bit of time for a breather in between the last lecture video and this one, just to give your mind a minute to process all of that information again, because we will be diving here into likely net new information for you, talking all about sequencing databases. So, databases are a way for us to provide structure when we're storing large amounts of data. My question for y'all is to try to think about why might we need this in bioinformatics and why couldn't we just use text files in software like Microsoft Excel instead? I'm going to leave that as an open question. I'd like to have that discussion on the discussion forum. So put your answer in underneath the header that I'll make for that in the course questions section. Databases are a way for us to logically organize collections of data. So we're typically going to be using these things called records to represent data within our database. And records are just a way for us to specify what kind of attributes we want stored with a specific kind of data. So you can literally think of them as like actual records that you would get from a record store that have music on them and things like this. That record is storing a collection of music, but it also contains attributes like the band name, the track list, the record name, different things like that. Just like with that, we want to use something similar when we're storing biological data. We don't just want a whole bunch of collections of letters all over the place with no information as to how these came to be, what they're related to, what organism did they come from, etc., etc. So a record is going to be a specific instance of a record type, but the record type, all records within a database would contain. That might be a little confusing. It will get clear as mud as we work through this lecture. So the very first thing we're going to do together here, I won't make you do this one alone and post on the discussion board, is conceptualizing what a record could look like for vet clinic patient information, and then y'all can do a different one for student information. So we know I got 2 dogs. They go to the vet quite a bit. And the things that I'm often asked when I'm going to the vet are things like, what's your animal's name? What kind of animal is it? What's your name? What's your phone number? Do your animals have any kind of allergies? So these are all things that they're collecting as information from the pet's parent or from things like the pet themselves that they are going to be storing in their own internal database at the vet. So there's going to be things like, I'll just do this, patient information. There's going to be typos and that's going to be our record type. Inside of this, we're going to, whoa, as I tabbed over, what happened? Here it is. Inside of this, we're going to want a whole bunch of different attributes. So when I'm thinking about this, I'm thinking about things like our pet name, pet type, owner name, owner contact, maybe allergies, things like that. This is not a comprehensive list of everything we could include inside of a pet record for a clinic. There are multiple different answers that would suit this, but we just need to think about what kinds of information would they need. So if you're coming up with things that I don't have on this list, it's very possible that they are also correct. You can post in the discussion board if you'd like some validation there, and I'd be happy to give you that. But that's what I'm thinking mostly when I'm thinking about this patient information. Maybe they would like to keep some payment information on hand. Maybe if the pet has had testing before, like x-rays or blood work, they want to keep records of that as well. Different things like that. One thing that this record is missing is a unique identifier. So right now, all of these different things, there could be more than one record with the same information. So I would think about, say if I was putting this together for my pets. So, why is that so small down there? I would have the Vader information. So for patient information, my name would be Vader. My pet type would be French Bulldog. owner name is Kim, owner contact. Y'all know how to get a hold of me. Let's just put some numbers in here. What would be my phone number, say? And Vader's allergies is pretty much to everything. So instead of listing them all, I'll put them like that here. But I'd also have another record at my vet for Rambo. So that record would contain Rambo, would contain French Bulldog. Owner name would still be Kim. Owner contact would still be whatever random number I decided. That's not the same. There we go. And Rambo is not allergic to anything. So he would have maybe an NA or something like that in his allergies. So if I'm looking at just these two records, if I use my pet name or the allergies, I would be able to distinguish the two of them. But I can guarantee you that there are other pets with the name Rambo and there are other pets with the name Vader. So we couldn't use that as a unique identifier for every single record within that collection. Similarly, for the allergies, they might be different in these two instances, but there's lots of dogs that don't have allergies, and there's lots of dogs that are allergic to everything. Like, I think Vader's like allergic to grass or something silly like that, right? So there are more dogs than just him that are allergic to that. So right now, there's no way for us to uniquely identify this. Even if we think back to, say, our actual music records, so inside of a record store, there's lots of different music records, and the way that they would distinguish between them is usually things like the SKU or the barcode that's actually on that record, so that product number. So in this case here, if we can't think of something that's like really easily attainable in terms of a uniquely identifying thing, oftentimes we'll just want to add an ID. So that could be something that we add right at the beginning, or here, I'll just put it at the end because I'm not typing everything down. Could be something like a patient ID. And that would be a sign when the record is made, say Vader is patient 2, and maybe Rambo is patient 576, or whatever it might be. Could just be a number that gets incremented every time there's a new patient that gets added into the system at the vet clinic. When you're doing the student information one, you're thinking of like, oh, what kinds of information might be relevant in a student record? Think about what types of things we have at the University of Saskatchewan that already are associated with you as a student that could be used for that ID, that unique identifier that is required for that database. So oftentimes the unique identifier is going to be called a key or an index. Very often it's like a number or something like that where we're just increasing that number every time we have a new record added into our database. We need to be very cautious when we are choosing what key we're going to be using to distinguish between records because they have to be unique. So you cannot have two that have the same. Because if we were then trying to search and we said we found this wonderful record too, but then it was also assigned to some other record with different data, how would we know what data we are talking about? So it's super, super important that those are unique. Numbers are typically the easiest because they're unique, but it could also be some combination of letters and numbers, whatever would make sense. And we'll see some examples of that when we look at the biological databases. So a database management system is a program that's going to enable us to create and control a digital database. It is like a step above Excel. So you know how in Excel you can have different sheets that contain information or different cells that are filled with information. It's this collection of information. A database management system is kind of like Excel in that it's collecting all of this information, but it's also allowing us to provide guidelines around what information we need in different kinds of records and a way to easily search through all of the different kinds of records that we have. So it's serving as an interface for searching as well as a kind of restriction or double checking when things are getting put in that they're containing the different fields that we said are important. Microsoft Access is an example of a database management system. So it looks a lot like Excel when you open it up if you have it on your computer, but it is slightly different in terms of how it's structured and the different rules and regulations that you can apply to data in it. So you'll see here that this is just a very simple database that has first names and email addresses. So one row in this, kind of like an Excel sheet, but it's a little bit different. It is Microsoft Access sheet, is a record. So it has all three of these different attributes. Each column is a different kind of attribute. And then the very first column we have here is our unique identifier. So that would be our key or our index. And you can see that it is just a number that gets increased as a new record is added. And this down here, this auto number, if you're ever using Microsoft Access, just tells the computer to automatically make that number for us so that we don't have to keep track of it, which is really, really nice. We have so many databases for biological information, whether that's sequence information for DNA, for RNA, for proteins. That might be protein structure information. It could be some of these next-gen sequencing experiments that we'll talk about later. We have lots and lots of databases available to store this information so that researchers across the world can leverage things that have already been done instead of having to redo them themselves when they're looking at, say, a new organism or a new disease, etc., etc. There are three major sequencing databases that we'll talk about in this course. So GenBank is the one that is in the United States. It is operated by the National Institute for Health, or NIH. The European Nucleotide Archive is in Europe, and it's operated by EBI. And then in Japan, they have the DNA Database of Japan. All three of these databases share information between each other. So they all contain the same information. And you might be asking, why the heck do we have three then? And it is actually a little wild why we have three. Because it's a digital database, all the data lives in a server at some location. If something were to happen at that location and the server went down, got destroyed, was damaged in a fire, maybe it overheated, things can happen. That information would all be lost if we didn't have some level of redundancy built into the system. And that level of redundancy that we've chosen as a community is to have three distinct locations in different parts of the world that all share the same information and contain the same information. So if something happened to NCBI in the States, that information wouldn't be lost forever. It would still be available through ENA in Europe or DDBJ in Japan. So it's just giving us a little bit of redundancy, a little protection in case something were to happen to one of those physical servers that actually is storing that data. When I'm talking about a server, a really easy way to think about what that actually kind of is, back in the old days, oftentimes like computers now are all integrated into like a thermometer and stuff, but we used to have those computer towers. If you're a gamer like me, you probably still have a tower that goes beside all your monitors and things like this that stores everything that's in your computer and is what actually is displayed. These servers are effectively really, really large computer towers that have lots of available memory in them to store all of this different information that we were putting into these databases. So GenBank is the one we will typically use for this course because it is located in the States. It's the least amount of lag when we're searching and different things like that. It currently holds over 1,562 billion nucleotides. And that was done in 2022, that stat. So I probably should update it. It's probably significantly different. I feel like it might be really interesting for you on your own to go and find out what that number is to see how much we add, like in the last just even 2 years, how much has been added, because it is wild. You can access it through this website here, which is the NCBI website, or through what we call, well, some people call it Entras, some people call it Entras, whatever, through this other link down here. This link down here, though, I just tried it, and it doesn't work. And the reason it doesn't work is because they changed it, and, okay, what? Oh, goodness, it's trying to click on things for me. Well, that's wild. Just get this. This part here doesn't exist. It's just /search now. It's not /NCBI search. So if you're trying to click on that, it's not working. Just remove the NCBI and then it will open up. When it does that, you should see, let me just switch this here. Something that looks like this. So, ooh, and I feel like this is not very zoomed in. Let me try to get this more zoomed so that it's easier to see. Hopefully that's a bit better. Yeah, that's a bit better. So we have a search bar here. We have some links to the various databases that this would be searching. We're going to get into more and more of these, so don't be overwhelmed by all of the different ones you see here. We'll go through some of the important ones. We won't go through them all, but it gives you a little insight into what Entrust looks like. So NCBI itself, if you clicked on the first link, it looks like this. So it also has a whole bunch of databases. You can search up here. You can specify what database you want, and we'll play around with all of that in a little bit here. Oh, I wish this would just keep my things clicked that I wanted clicked. That's okay. All right. So Entrust has about 40 biological databases that it will search and retrieve information from. It's going to integrate with all of the major DNA and protein sequence databases, along with taxonomy databases, genome databases, protein structure databases, mapping databases, journal databases, you name it, it's there. It's a really good initial kind of place to check when you're looking for new information. Oh, see, this one has the right link. So we're going to go to this link and we're going to search for this gene symbol here called HBB. So gene symbols are typically 3 letters long, all capitals, and we're going to see what we get from that search. So going to this site here, so remember we have the search at the top, and we're going to be looking for, what was it again? HBB. HBB. So we'll just put that in, hit enter, or press search, and we'll see what we get. So the very first thing we get up here is this information that, hey, yeah, this is a gene. Your prof does know what she's talking about some of the time. 3 letters are typically representing gene symbols. And this one in specific is looking at the hemoglobin subunit beta. So up here, we could click on this, and we will click on this, and we'll see what comes up there. But you'll see over on the side, we have these things called RefSec transcripts. You won't know what those are yet, probably. We'll get into it later in this lecture. And if we scroll down, we can see things like journal articles that are related, gene datasets that are related, protein datasets that are related, clinical trial information that's related, any kind of genome information that's related. It's all kind of here, which is really, really nice. So if we click up here, this will load what we call the gene page for that gene that we were looking for. So it's going to contain a whole bunch of information on here, and we're going to go through this in more detail together as we go through the rest of the slides. But you'll see here is our gene symbol that we have. Whoops, as I click on other things, my goodness. It has summaries here. It has lineages for the phylogenetics that generated it. It has what organism this is coming from. If we scroll down, it shows us where our exons are. So remember we talked about how some have introns, some genes have introns that need to be spliced out. This gives us the locations of the different exons if we wanted those. We have some visual information down here that's very overwhelming when you look at it at the beginning, but we'll break it down together and we'll say, okay, what do all these different tracks here mean, as well as things like expression and relevant journal articles and things like that. So you can feel free to click through this, see what you find. If you have any questions, as always, just let me know. I'm here to help. Let's pop back here to the slides. Here we go. So when we are searching things like the Entrust database, every record is going to have what we call an accession number. So that's what we call keys for databases in bioinformatics, is accession numbers. Accession numbers are shared between all 3 of those major databases. So NCBI, DDBJ, and ENA. They can be updated. So say we get new sequence information that's more accurate, we're going to update that accession number. And when we do that, we add these dots and then the version at the end of it. So that's how you know what accession number you are. So this is an example of an accession number. And then the version here that is updated would be that .4. Searching for an accession number without the version will just take you to the newest version. So sometimes the versioning is important if we're trying to recreate method or results from a previous journal article and that record has been updated. We want to use the record that matches when that journal article was released in order to recreate those results correctly. There are some rules associated with accession numbers because they have to be unique, so we need to have rules around them. Nucleotide accession numbers are going to have 1 letter followed by 5 numbers, or 2 letters followed by 6 numbers. And you're like, do I have to memorize that? You'll just get used to this over the course of this of Bioinformatics 151. You will get used to it. You will see it. Protein accession numbers are database dependent. So sometimes, depending on the database that they're in, they'll have the abbreviation for the database at the beginning followed by some numbers. So SwissProt is a very common protein database. Protein sequences that were submitted there usually have SP before them, followed by some set of numbers. So we'll see lots of different examples of that. So now we're going to move on to exercise 2. I'm going to show you what it will look like, and we'll walk through this together because we haven't done this before. So instead of doing it on your own, we'll walk through it together. What I would recommend trying, though, is pausing the video here, trying this on your own, and then watching it to see, oh, okay, where did we find this information versus that information, etc., etc. So we're going to be looking for NM000518 in this nucleotide database. So you can click on this link here to go right to the nucleotide database. I am going to click it open. Then I'll switch out into that window. And I just want to I'm going to copy pasta that accession number from the slides so that I don't have to try to type it out and remember. Oh, and my copy pasta isn't working. There we go. So up at the top here, you'll see it says, oh, I should go back. Up at the top, you'll see you're going in the right spot because it says nucleotide. Right beside that is where you would enter that accession number. So if you've just pasted it over, or you can type it, it's NM in capitals _000518. And then all you need to do after that is hit search. It should load up. Hey, this looks familiar. The exact same page we initially searched. So that gene HBB, this is the same thing. But in this case, it is the mRNA sequence, not the full gene sequence. So the first question we had was, what organism is this from? So we have a whole bunch of information in here. Like if we scroll, oh my goodness, so much information. And we need to find where the organism that this is from is located on this page. Conveniently, it's always going to be in the same format here. So the same information will be listed in the same sections. And in this case, what we're looking for is source. So source tells us the source organism that this sequence is from. And in this case, it is Homo sapiens or human. The next question we have is, what is the length of this sequence? So there's multiple places on this page where you can find that. The easiest place to find it is actually right at the top here on this locus line. So that gives us our accession number, and then right after that, it gives us the length of the sequence in base pairs. We can also scroll all the way down to where we have things like our features section. So that's going to also tell us for each of these different features that we have in here. So for the gene, the exon, miscellaneous features, which base pairs those features contain. And at the very top, it should always have the source, which should be the whole length of the sequence. So you'll see it goes from 1 to 628. So our length should be 628. And that does match that one up top, if I'm correct. Yes. Oh, the anxiety. It just never goes away sometimes. So you see there's lots and lots of different features that we can come on here. You'll see in the labs and things like that, we'll be asking you to search for the different things on the nucleotide website. And we'll be asking you things like, where is the exon located? So you'd scroll down and you'd find that base pair spread, and that would be your answer to that. Or where is the coding sequence, which is the CDS? And that would tell you this right here. The next question we had, and the final question, is what is this protein that the sequence codes for? So we get a hint up here that it probably has to do with HBB, but how could we be sure that that is what it is? So we know this is related, the mRNA related to the hemoglobin subunit beta, or HBB. So that is for sure the protein that this is coding for. But we should also be able to scroll all the way down to where we were at the coding sequence that we were here, the CDS, you'll see in that section, after it gives us the base pairs of where that coding sequence is, it also tells us what gene it is, what protein product it gives us, the accession for that protein product, and even the translation for that protein product. So it gives us all that information we could ever want right here. So we can get the protein ID, we can get the gene symbol, we know the product is hemoglobin subunit beta. You can even grab the sequence from there without having to click into anything else, which is super, super nice. So if you have questions about that, let me know. You will get more practice with that in the labs and the assignment as well. So there are lots of different types of sequence databases, but generally we will categorize them into two distinct types. So primary sequence databases store experimental results. So they're just, if you've run an experiment, you've published something, you need to typically submit your sequences to some sort of database. These primary databases are what are going to store those results. So they'll give you an accession number that you can link into your journal article so that other people can use that same sequence. Secondary sequence databases are curated. So they often combine multiple records into a single curated record. So they'll try to get a representation of all of the sequences that have come in for, say, HBB, and get a representative sequence for that. So it will be pared down into one individual sequence. And we'll see some examples of these secondary databases later on in this lecture. The primary, I shouldn't say that. The main one that we will use as a secondary sequence database in this course is going to be RefSeq, but we'll get into that in a little bit here. Within sequence databases, we have different kinds of file formats for our records. Oftentimes we'll be doing or using things like flat files, which is usually just a text file. Think of a flat file as just a text file, just a whole bunch of text that we can then go in and process. So there's things like the GenBank format or things like the FASTA format, and we'll see examples of both of those. And then also sometimes records have graphical formats. So we saw that when we first searched HBB and we scrolled down and there was like that wild image with all these different tracks that could mean different things. It was very graphical and very interactive. So we'll see examples of that as well. If we want to see the FASTA format, all we need to do within the nucleotide database when we've searched it is click FASTA. So I'm going to actually just switch over to that nucleotide database search again, and then I will go through the slides on the database instead of the individual slides itself. So you can have those up on the side if you want to follow along, but we're going to just click through in this different way. So right now here, we're in GenBank format. We can actually save this to a file by just saying send to, we would say complete record, and then we click file, and we would download that one GenBank format and create that file. That will just download it. It will give it the extension .gb. So if you wanted to open that, you need to open it in a text format or text file. So something like a notepad will open that. If you're on Windows, I'd highly recommend downloading like Notepad++. It's really great for coding as well as opening things like .gb files or .fasta files. If you're on Mac, things like TextEdit or Text Wrangler are really good for that. So this is literally just an exact copy as if we were to like copy paste all of this text into a file. So it has a whole bunch of extra information up here about things like different accession numbers, our version, so we're on version 5, organisms it came from, papers it's related to, all of these different features it has, etc., etc. If we only wanted the sequence information, what we would want to do is switch this to FASTA format. So FASTA format is the format we typically use in bioinformatics to store sequence information. And there's two ways you can get that on NCBI. You can just click this FASTA button here, and that will load it up, and this is your FASTA format that you would see. Or if we go back to the other page, up at the top where it says GenBank here on the top left, you can click that, and you can click FASTA, and then it will load that as well. So this is just our FASTA format. And things we'll often see with the FASTA format is you always have this greater than sign starting off our header line, followed by our accession number, maybe our organism if that information is available, and then a brief description of what the sequence is. So in this case, it's the hemoglobin subunit beta mRNA. After that, we just get lines and lines of sequence information. So A's, T's, C's, and G's if we're in DNA, amino acids if we're in protein, U's instead of T's if we're in a true RNA sequence. Oftentimes, so we do know that this is mRNA, so that just means that there is no introns in it, but you'll see there still is T's here. So this is the mRNA sequence, but we represent it oftentimes in the databases with the T's instead of the U's. Very confusing. I apologize. I didn't make the rules. I just live by them. You can also save this this format to your computer by clicking Send To up in the top right. And then you just click File again. You'll download the FASTA format and create the file. And that will create something called sequence.fasta. So that .fasta type, again, can be opened in any kind of text editor. And it will have the same information as if we just copy pasted this into a file itself. The other thing I forgot to mention about FASTA files is that complete lines of sequence information will only have 60 nucleotides. So you'll see we always have this line break here, and then our last line is usually less than, so some amount smaller. We do that so that it's easier to view on a page or in a console, different things like that. So we always know we have those 60 nucleotides, and then it will be a new line, and it will continue on from there. One of the questions I have for y'all to think about and maybe post in the discussion board is, what are the benefits of a FASTA file? Why would we want to store our data in this kind of way? When you think about this from a bioinformatics perspective, what advantage would having our data like this give over us having our data in something like the GenBank format, like this? What benefits would that be? Would it be easier to process? In what way? Does it still give us the information we need? I'm open to your thoughts. Let's have that discussion. So if we go back to the GenBank sequence here, you'll see here we always have our identifying information at the top, which I mentioned, followed by any kind of features that we have here. And then if we keep on scrolling, we eventually get to our sequence here, which has at the beginning of each line the base pair that it starts with. And then the GenBank format always ends with a //. So yes, GenBank format does have sequence information in it as well. But if we had this versus the FASTA file, and say you needed to do a whole bunch of processing with that sequence information, which one would you prefer and why? We're going to now look at the GenBank file for a record or accession AH002877. So here, I'm going to just copy that from the slides. Again, I'm going through the slides, but I'm trying to stay on the website so it's just a little more straightforward, not so much back and forth. But you can follow along with the slides on your own. So we'll just paste in that accession numbers. Again, we have AH002877. And if we just hit search, let's see what we get. We get one record that came up, so we didn't have to click on anything. It knew exactly what we were talking about because we put that accession in, and it's only ever going to have one valid response for that accession. In this case, it's the myoglobin gene, and it's the complete coding sequence. So if we look here, we know where it's coming from, we know what organism it's coming from, we know how long that sequence is, and we can go down to the features and see all of this different information about where our introns are and where our exons are, and there's our sequence, etc., etc. This sequence is much longer than the last one we saw, but I'm wondering if you were just given this features information, how would you go about drawing a diagram of that gene where the introns and the exons are? So oftentimes what we would do is we would go in and we would look and we'd say, okay, so we have these repeat regions, etc. We don't need to worry about that. Our coding region is going to be all of these different places here. So what this join means is that it actually has 1, 2, as I'm searching, 3 exons. So the exons are the coding part of the sequence. And in this case, it gives you the base pair locations of each of those exons all in one. So you know, if you were to draw it out, you could draw a line, you could draw your little boxes for your exons, and you can say, this one starts at this base pair, ends at this base pair. Then there's an intron, and then the next one starts at 4067, ends at 4289. And we can double check and confirm that by going a little bit further down into the feature section, and we'll see our very first exon here shows up at 1965 and ends at 2059. That's the exact same as that first region that we have in the join, which is exactly what we want. Then we would see where the intron goes to, which is the next little interspacing thing. We have a couple of different things that are added after the intron, so we wouldn't necessarily need to like break all of that down, just to keep it a little bit more simple and high level of a diagram, but we can use all of this information to draw out a diagram of the gene. I'd like you to try that on your own. If you have problems, come to labs, come to help hours, ask questions in the discussion board. We would be happy to help you through that. Within these files, we also have a graphics format. So we've talked about the FASTA format, we've talked about the GenBank format, but we've not talked about the graphics format. If we click on that, it's going to be a little bit different. This is that big screen that we've seen before in that initial version. So your picture that you drew that lays out all the different information of where these different regions are should be kind of similar, but probably simpler than what we see here. So up at the top, we have all of our base pairs for this sequence. So we have 1 all the way to 6,889. So it shows it up right here. And within that, we break it down into different tracks that show the different features that were just in that flat file before. So this is a way that could help humans see this data. This is not useful for computers. Computers would way prefer the text-based version of this so that we could then go through and process and do what we want with this. Humans typically like to see things kind of laid out in front of us and interactive. So in this here, we see here's our whole gene. We have our different exons at these different locations and our introns interspaced in between them. We also have some repeat regions that have been found by the scientists that have done this research. So we can click on that. We can find more information about that if we wanted to. Oh, why is it not staying up? I'm using Microsoft Edge, which is probably the jankiest of the browsers for this. So it's not working perfectly, but if you click on it, you should get a little pop-up here that you see it goes away really quickly on mine, but if you're on Chrome or something else, it shouldn't. It gives you more information as to what all of these are. We have various introns, so we can click on that. We can get more information about the different things down here. So our accession number, which version that specific feature came from. We also have different things over here like the mRNA, so exactly where that would line up within the sequence that we have, etc., etc. So depending on how well researched that sequence is, we're going to have more different tracks here. I am honestly so used to the text version that I look at this and I feel like this is overwhelming. But I have to remember that I've been in bioinformatics for quite a number of years now. So when I started, I also probably preferred this version instead of just looking at a wall of text. But one day you might be in the same boat as me. So all of these different things that you can click on should also hyperlink out to various things at NCBI. So if you wanted more information on like a specific exon, or you wanted just that, you should be able to go and find it with the various hyperlinks that it will give you when it pops up there. You can also get information for each of the tracks by clicking on the little eye on the side. You can remove tracks from the view or hide them by clicking the X. And you can also download all the track data, but if you had like the GenBank format, you wouldn't need to do that. All right. So we're going to switch back to the slides now. I think I was just double checking that we were done with all of the things that I wanted to show you on the searches. So that went through effectively all of that file formats section. So we're up now into curated curated databases. So these are things like our secondary databases. So as I mentioned before, our secondary databases, the goal of them is to take all these sequences that get generated in our primary databases and pare them down into these reference sequences. So within this, oh, and that's kind of a hard screenshot to see. I wonder if I can zoom in here. Oh yeah. This is great. Okay, but then I have to figure out how to scroll on this thing. Please hold. Ah! The computer is trying to be helpful and resize my screen for me, and it's like, that is not what I want. But I will resize it just for a second here so I can get it all in one. So if we go over here, as I mentioned, RefSec is one of the most common secondary databases. Things like GenBank would be a primary database. So you see, if we look over at the side here, when we did the search for myoglobin, we had 261 records come out in all of GenBank. So that's for all the different organisms. There's going to be redundancy in there when multiple experiments have been done on the same organism, so they have similar sequences for similar regions and similar organisms. RefSec has far fewer records, only 84. And the reason for that is because if, say, we had 13 human sequences for myoglobin, RefSec would take all 13 of those sequences and combine them into one reference sequence. So its goal is trying to give us a reference sequence that represents all the data that's in these primary databases. Can I zoom out? Why is this like that? There we go. All right. So the goal of these secondary databases are to curate. So you'll hear that term a lot when we talk about them. The curation process is sometimes automatic, but a lot of times involves manual intervention by people to go in and say, does it make sense to put these two things together? For instance, if you had sequences related to a disease like sickle cell anemia, you wouldn't want to just combine all of the human hemoglobin sequences together. You might want to separate those out into groups where they share similar phenotypic characteristics when it comes to that particular disease, stuff like that. So the goal is to provide the representative sequence in RefSec for each quote-unquote normal gene, transcript, and protein. You'll learn as you go on in bioinformatics that normal is a weird word that we sometimes use in bioinformatics. Typically, it just means not in a diseased state, not from a specific phenotype, represents the average population, that kind of thing. I don't have a better word for it than normal right now, but it does give me the ick every time I have to use that. Because what is normal? NCBI will often provide us a shortcut to RefSec information when we search. So if you searched up the gene for myoglobin, which you could easily do now, I'm confident about that on your own, you'll see that we have things like RefSec transcripts, proteins, genes, all linked at the very top. And then we also have the individual transcripts for RefSec at the bottom here. You'll know that you're dealing with a RefSec transcript or accession number, when you have those two letters at the beginning followed by the 6 numbers after. So each of those 2 letters at the beginning is going to represent a different type of sequence. So NC RefSec accession numbers represent complete chromosomes, genomes, or organelles. NG accession numbers represent incomplete genomic regions. NM represent mRNA sequences, and NP represent protein sequences. So you'll see we'll do a lot of work in this class with the mRNA sequences in RefSec and the protein sequences in RefSec. So you'll see those come up time and time again. In Entra's Gene, we have also a curated database containing information regarding various genetic loci. You can click on this link to open it up. So this is curated. Unlike the Entras we were searching before, this is specifically related to genes. So if you open this up here, you should get a page that looks like this. You'll know you're on the gene one because you have gene selected here, and you'd be able to look up gene information on here and get curated information. So let's try this using the ACE2 gene. So all we need to do is search ACE2, hit search. See how fast my internet works? You'll see we have our curated record up here for gene. If we click on that, as long as we want the Homo sapiens one, we get all of this information that has been curated for us. So we have our gene symbol, very similar idea to like the GenBank format we've seen before. Our primary source, the name, if it's been reviewed, what kind, is it a protein coding one, etc. A whole bunch of summaries related to this. You scroll down, we get exons, assembly reference information, visual descriptions of what's going on for that gene. And you'll see here in this track here, we have so many different RefSec sequences that are related to these things. So we could click on that, hover over it. We could go click on a link to get that gene sequence that's related to it. We have things like promoters and transcriptional regions, all demonstrated in here. So there's lots and lots of information that we would see in this section here. We also have protein sequences in NCBI that have been translated from nucleotide records in NCBI. So one of the things that I mentioned before that we often do in bioinformatics is take a DNA sequence and translate that into a protein sequence. So those translated sequences do exist in GenBank, but there's also external databases that just contain protein sequences. Those ones are typically things like the Protein Data Bank, SwissProt, UniProt, etc. We'll see more examples of those later on. There's some more resources if you want to read more about this. There's a textbook that's in the library by Moment and McCurdy that you can read. So is this one by Christiania and Han. So if you want the names of those, I'm happy to provide them. There's also some how-to guides on NCBI, which I would recommend more, that would give you information about how to do all these searches if you're still a little bit lost. And of course, you can just ask questions and I will help you out as well. There are some tricks we can use for searching for particular things on NCBI. So if we put things in quotes, it will search for a whole phrase instead of just the individual words itself. These tricks can also be used on Google, so you might know them already. But so if we wanted to search for the contactin-associated protein, if we put that in a quote, it would search for that as a collection of all those three words together instead of each of those individual words separately. There's also logical expressions that we can use to build up more advanced queries. So if you want two things to be true, say you wanted the hemoglobin gene and you wanted it in dogs, you could use that "and" keyword in all capital letters to combine those two things together. If you wanted all of the sequences that weren't in a specific organism, say you weren't in humans, you could just add "not" in capital letters there, humans at the end, and that will restrict any human results from being returned. You can also use "ors" to give if you want one thing or another thing. Maybe you want flies or dogs in your organisms. You'd be able to use that to build them up. And we can use brackets just like we do in mathematical expressions to nest things and add importance to other things in order of operations, etc. So there's lots of different things we can do there. You'll get lots of practice with that in the labs. There's also some query language expressions that are specific for NCBI. So there are some quick tricks that we can put here. If you want to restrict it to only RefSec, you can add this "and RefSec" filter. You just copy and paste that right after your query and it will only return RefSec results. If you only wanted it to be Homo sapien results, you could add this one "and Homo sapiens organism" and it would restrict it to that. So you'll get a practice with this in the lab, so we won't do too many examples of that right now. On the side here, when you are searching, you will see those search details from the queries being built up here. So you can use that and edit that directly and then make changes and search again, and it will update your results. So say you wanted to change it from Homo sapiens to a different organism, you could easily do that in there. Just make the change and then hit search again. You can also click through to restrict and it will build that up for you. The clicking through takes longer, so using these tricks will save you time, but both will get you the same results at the end of the day. That's all we're going to be doing this week in the class. I hope y'all have. Oh, I guess not. You can tell that I need a break. That's the end of this lecture. It's all we'll be doing in the week 2 here of this class. So I hope you enjoyed these first 2 lectures. Please reach out if you have any questions, and we will see y'all next week. Bye.